So, hello everybody. Welcome to our Usomi webinar. At this time, it's on radiomics. My name is Daniel Pinto. I'm a member of the board from Usomi, and today we are happy to have Laure Fournier with us. She's associate professor in Paris, and she works in the Laboratoire de Recherche en Imagerie. And she's working on radiomics, and she's an expert in this field. So we are happy to have her, and I'll pass the mic.
Thank you very much. So hello, everyone. Thank you for joining this webinar on radiomics. So you should be seeing my screen now. I just wanted to underline the fact that the things that I would show you are the result of work I've been doing with a lot of people, medical informatics people, with PhD students, residents, etc. And so, of course, this is the fruit of everybody's work. So I'll just jump right into the subject. If I can change, this is my disclosure slide. So I just wanted to disclose that I was collaborating with some industry and specifically on radiomics work.
May I just jump in? Sorry, I just want to point out that all the participants can make their questions in our Q&A section and use the chat to get in contact with you. And I think they should be free to ask you once there is a question coming up.
Actually, if it's what we would like to do is that the questions are at the end of the presentation, because yeah, that's how we suggested it would work. So to you all participants, if you have questions, just jot them down on the side. And then when we get to the end, I will, of course, answer any of your questions. We can go back on slides if you need to, so that we can go back to something. So to start the presentation, I want to start with the way that we were taught to do research, which is called hypothesis-driven research. This is what we learn when we go to school, which is that we usually have an idea, and so we're going to formulate a hypothesis, and then we will collect data specifically to answer this hypothesis. We will analyze it, we will interpret it, and we will see if our hypothesis was right or wrong. And this will lead, when we do imaging research, to hopefully the discovery of a new imaging biomarker. And of course, this biomarker will not be, I call it a candidate biomarker because it will have to go through validation. And there are two steps that we can kind of individualize in this validation, which is a technical validation where we will check if we have a robust biomarker that we can measure consistently the same way. And then there will be a clinical validation, which will regard rather how we can use it and whether it does reflect a clinical outcome or a disease or whichever we decided. So that's what we usually do. The limits of this strategy is that that means that every time you have an idea, you have to put together a project and you have to do this workflow. And so it ends up being very expensive and time consuming. That's the first limit of this strategy. The other aspect that I wanted to underline is that we are kind of changing our way of regarding images. And when you're an imager, you instinctively know that images are actually a phenotype. That means that whatever's happening, as you can see on the schematic, whatever is happening at the genetic or biological level will be reflected in your image. And so behind this, there was the idea that there was probably a lot more information in the images than what we're using currently. Of course, in clinical routine, we don't use that much information about our images. A few things about signal on MRI, a few things about size, of course, anatomical information, but not that much more information. So from this, this concept, there's been a real paradigm shift. And people have been moving to data-driven research. And the idea behind data-driven research is that we're going to turn the whole process around, and we're going to start with the data. So either you collect it or you have collected it and it's just sitting in there in your packs. And from this data, you're going to try to generate new hypotheses. So you're going to take this data, analyze and interpret it, and generate a new hypothesis. So you don't have it ahead of time. And what's interesting also in this data-driven research is that you can implement the qualification of your candidate biomarker during this process. And I'll illustrate that when I explain how we do radiomics. And so in the end, you will have a new candidate biomarker, and you will then, of course, still have to validate it clinically. So the other message of this slide is also that it doesn't change anything. You discover biomarkers, but you still have to validate them. So what is radiomics? Radiomics is a data-driven research. It's the extraction of a large number of quantitative parameters. This can be done in an automated way with this automated segmentation or manually with no a priori hypothesis. And then once you've extracted this very large number of quantitative parameters, you will want to correlate it to something. So it might be outcome, or it might be a gene expression profile, for example. And in that case, you would call it radiogenomics. But once again, you have to understand that this is discovery research. It's not a validation. It's not because you discovered something with radiomics that it's meaningful, but it will generate new biomarkers. You will discover new biomarkers that you might not have thought of yourself, and that way then you can go on to validate them. So it's kind of like this old man that's panning for gold in the river. You're going to go through a very large number of parameters. A lot of them will just be, you know, little pebbles, but you might actually get a gold nugget in there. So what is the process? You can individualize 3 steps, basically. The first step will be collecting the data, of course, and you can do this by image processing. So you will be extracting features or parameters. You can do this in a traditional way, or you can use artificial intelligence. So artificial intelligence is not the same thing as radiomics, but you can use artificial intelligence in radiomics. And what's interesting in this process is that you can extract these features from routine clinical images. You don't have to do a special acquisition as you might if you wanted to do perfusion imaging, for example. So you usually start with very routine images. The second part of the process is the worst, the most boring, and actually the one that will often limit you in a way, which is the cleaning of the data. It's called data curation, and that's when you check the quality of your data. You make sure that it's very uniform. You might enrich it, and at that stage, you might want to add genetic information, biological information, clinical information. So you can actually add information that doesn't strictly come from the images at this stage. But unfortunately, often that's where you lose a lot of your patients, because since you're working on routine images that you acquired retrospectively, well, all of them are not, you don't have the data that you need for all of them. So the third step is the data processing step. And so you have a very large number of features. You're going to have to deal with these features and correlate them to something. And so usually it's you do it with medical informatics and biostatistics methods. And at this stage, you can also use artificial intelligence to choose which are the most significant features. The process that I'm going to show you does not use artificial intelligence. It's the very traditional process, which is a 2-step process, that has first reduction of the number of features, and you can do this based on the technical quality of the features. So that was the idea I was telling you about of incorporating technical validation during the radiomics workflow. And the second part will be the one that we're most interested in most of the time, but it only happens at the end, of course, which is test it for the clinical relevance. Okay, so let's take each of these steps one by one, and we're going to go through them. So the first step we talked about was extracting all the very large number of features. So the way you're going to do it is you're going to have a patient population with CT scans or MRIs or PET or whichever imaging technique that you would like. You can actually even do it on photography if you want. And you will delineate your region of interest. I work on cancer, particularly renal cancer. So the image that you're seeing is actually a primitive renal tumor. So you're going to draw the region of interest. Once again, you can do this automatically, or you can do it manually. From this region of interest, you're going to have a series of pixels and you're going to process to extract features. So before this, you might have to apply filters or aggregation. I won't go into that. If you have questions about that, we can talk about it at the end. There are three categories of features that are individualized. There's shape, signal distribution, and texture. So shape will be basically, I think it's self-explanatory. You will describe what the tumor is like, how the size, of course, its surface, its volume, how compact it is, if the contours are regular or irregular. And so there are a lot of mathematical ways to calculate these features about the shape. The second part will be signal distribution, and this is also called histogram-derived parameters. So basically you just display on a histogram the number of pixels that exhibit a certain signal intensity. And then we know how to quantify things from histograms. We know mean and standard deviation, but there are also new parameters that we've been having to get used to, which are skewness. And you can see at the bottom on the graph, the skewness is whether your distribution tilts to the left or tilts to the right. And kurtosis, which is whether this distribution is very pointy or not. And then there are very more complicated parameters such as energy, entropy, which are harder to explain just on a graph. The third class of features are texture-related features, because the problem with the histogram is that you know what kind of pixels are in your image, but you don't know their relationship to each other. And what's interesting about texture-related features is that they're interested in how the pixels are distributed spatially. So basically what it means is I want to know if a pixel is surrounded by pixels that are the same or pixels that are different. And you understand that what texture actually quantifies is heterogeneity. And everyone wants to know about heterogeneity, particularly in tumors, because we know this correlates to its aggressiveness. So the way it's done is by calculating a matrix, and I'm not going to go into it, but basically the matrix describes what I was saying is how many pixels, for example, in a row are the same value, or in diagonally, or in circles, or whatever. But that's the main message. So once you have all these different types of features, you're going to end up with a very large number of features. So what does very large number mean? It means at least more than 100. Usually we're in the several hundreds, even a couple thousands. But the problem is that you usually have a lot more features than patients, of course, because in most of our medical studies, we usually have on the order of several tens of patients, not several thousands. So we have this very large number of features, and we also know that statistically, if you have a lot more parameters than samples or patients, then you will have a very high chance of making false discoveries. So the next step is the feature reduction or the feature selection. So the idea behind that is I have a lot of features, but I don't want to make false discoveries. So I want to get rid of a lot of those features that might not be interesting. And there are several ways to do this. The first way to do this is a kind of machine-driven way, where you will use either unsupervised approaches or supervised approaches to separate features according to their characteristics, let's say. The only one I'm really going to talk about is this clustering technique, which is hierarchical clustering. And the reason I'm talking about it is because it generates what you see on the right, which is a heat map. And these are things that in genetics, they do all the time for genomics research. And so I think it's interesting for us to understand what these heat maps are. So I wanted to take the time, sorry, I changed my slide. I wanted to take the time to explain how they computed these heat maps. So these heat maps are computed using clustering. And the idea behind this clustering is that you will represent together patients that have similar characteristics. I'm sorry, my slides are going through a bit faster than they're supposed to, so please excuse me. So if we represent a graph theoretically of 6 patients that are called A, B, C, etc. to F, The first thing you're going to do is you're going to measure the distance between them. So measuring the distance is seeing how different they are. So of course, if you only have one feature, you're going to say, patient A has a value of 6 and patient B has a value of 8. So the distance is 2. Of course, when you have lots of parameters, it's more complex, but basically it's the same idea. You're just going to put all the parameters together and say which 2 patients are the closest together. So you're going to measure the distance between each of these points. You're going to identify the closest pair, and then you're going to merge them. You're going to put them together, and you're going to represent them by the middle. Okay, and you're going to start over each time. So we're going to go through this. So imagine we have these 6 patients, A to F, and we've measured all the distances between each 2 points. So between A and B, A and C, etc. The ones that are the closest are D and E. Okay, their distance was measured by 0.94, and so we're going to represent them like this. You see it at the top right. You're going to write D and E are the same, and their distance is 0.94. And then you're going to go to the next one, and you're going to say A and B are the closest. So you're going to represent A and B, and also draw these 2 lines between them, and you see that these 2 lines are a little longer than the ones between D and E because they're further, etc. Then the middle of D and E is closer to F, A and B closer to C, and so etc., etc. And you get this clustering that tells you how far each point is to the other, okay? And then you can threshold this. You can say, I want 2 groups of patients. So if I put my threshold here, you will have a group that's A, B, and C, and a group that's D, E, and F. Or conversely, if I put my threshold here, I will have a group that's with 5 groups of patients. So that's the way it works. And in the end, you get this heat map, which is also color-coded. And so at the top, you will have the number of patients, you have the patients, and on vertically, you will have the features. And so the patients that are the closest to each other will be represented next to each other. On top of that, you have a color coding that gives the value of the features. So the ones that are high are in yellow, and the ones that are low are in blue. And that way, you will have a visual representation of your whole population. And you can separate them and say, visually, I can see 5 groups of patients that have features that resemble each other. Okay, and if you generate this heat map, you can then go back to your clinical data and say, okay, I have 5 groups of patients on my images. Do these patients have different clinical outcomes? Or do these patients have different genetic expression? And that's how you use these heat maps. I hope you guys followed this. So I'm going to go this is one of the way to do it, but it's not a very simple way and it's very exploratory. So I'm going to show you another way to do it, which is a very traditional way, and is less exploratory. What I mean by that is that in genomics, they've been doing this for years, and so we know how to use these techniques. So instead of doing the heat map, I'm not doing the heat map at all. I'm going to do a feature reduction based on feature quality. So remember I said, I'm going to discover a biomarker and I want it validated technically. That means that I'm not interested in discovering a new biomarker that would not be reproducible, for example. Because if I discover it and no one can measure it the same way, then I'm not interested. I don't even want to discover it. So what I'm going to do is I'm actually going to get rid of all the features that are not reproducible. So when we draw regions of interest to extract our features, I'm actually going to ask several radiologists to draw these regions of interest, and I'm actually going to ask one of them to do it several times so that I have an intra and inter-observer reproducibility for every single one of my features. And I'm going to get rid of all the ones that are not reproducible. So there are lots of ways of quantifying reproducibility. I just wrote 2 different correlation coefficients that we use. And then we threshold them and say everything that is not very highly reproducible, I'm just getting rid of it. The second way to, the second aspect that we would like to use for feature reduction is how significant or how relevant or how meaningful the features are. The idea behind that is that since we extract thousands of features, well, a lot of them will be highly correlated. For example, if you have a tumor which is a sphere, you know that its diameter, its surface, and its volume will be highly correlated. And measuring all three of them will not add any additional information. So instead of taking all three, I can just take one of them. So we do that using a Spearman correlation, which probably a lot of you have already done. That means we see how correlated each feature is 2 by 2. And then we put them together, and instead of keeping all the ones that are correlated, we just keep one of them. Okay, so these nice graphs that my PhD student made basically show that things that are very dark blue are very highly correlated, and those that are red are anti-correlated, but basically they all give the same information. So we get rid of them, and we only keep the ones that give specific information. So just to give you an example, one of the studies that we did in 2 different populations, and we extract practically 1,800 features from our images, from our CT images. And then if we select them for reproducibility, so here is represented the 2 populations, then we get rid of a lot of the features, and you'll notice that we don't have the same number of features that we got rid of in the 2 populations, because it really depends on the nature of your images and the acquisitions. So we get rid of those that aren't reproducible. Then we get rid of those that are redundant, that give the same information. So we replace those by a single one. And in the end, from those 1,800 features, we end up with a little over 20 features. And 20 is something we can deal with. We know how to deal with this statistically. And it is only this small number of features that we will use then to predict the clinical outcome. And just to show you the example in our study, we identified 3 features that are called lengthening, skewness, and GLN. And we could separate 2 populations of metastatic renal cell carcinoma patients, so renal cancer patients with very different progression-free survivals. One had 2.7 and the other one had 18.4. So you see there's a very big potential to discover things that are very significant. I'll just show you 2 examples of publications that I find illustrate how radiomics can be interesting. So these examples are in oncology, because that's what I work with. But of course, radiomics can be done in any kind of disease. This first paper is, I like it very much because it is actually one of the first papers on radiomics. It's actually radiogenomics, since it correlates the images to gene expression. So it was in one of the Nature journals in 2007. So you can see this is quite old. It's on hepatocellular carcinoma. They had a training set of 38 patients and a test set of 32 patients. So you see the numbers of patients are usually not very high. They had practically 140 imaging traits on CT, and a gene analysis of over 6,000 genes, and that's what they correlated. What's interesting is that among their imaging traits, there were some that were quantitative, that were calculated by image processing, but a lot of them were actually traits that the radiologist would identify visually, because even though we are interested in quantitative imaging, it's probably complementary to the visual assessment of a radiologist. And so it's interesting not to lose the information of the visual assessment and to include them in the radiomics analysis. What this slide shows, because it's a kind of complicated slide, is that they started with 138 imaging traits. And what they did is they correlated to the genetic expression. And they discovered that actually, if you took 32 imaging traits, you could describe every single genetic expression pattern that could be observed in HCC. And so you actually did not need all these traits to describe everything, but every different kind of patient could be identified if you combine these 32 traits. But that's not the thing that I was the most interested in. What I was most interested in is on this new slide, because started looking at what imaging parameters correlated which biological feature. And so I put a little square around the right part of the graph, where it shows which imaging traits correlate to genes that are linked to extracellular matrix remodeling. So this extracellular matrix, we know that when tumors grow, they invade it, and the more invasive they are, the more they will disseminate, of course. So if there are anomalies in the gene expression of the extracellular matrix, well, it will reflect on how invasive the tumor is. And what was really interesting, if you look at the top line on this slide, is that there's something written tumor margin score. And that was a visual evaluation of the tumor margin. So the limit between the tumor and the environment was very highly correlated to the genes that were implicated in extracellular matrix. So I find that really interesting to see that there's a logical link between a tumor margin that we might observe and the abnormalities of the extracellular matrix around the tumor. The second paper that I wanted to talk about is probably the best known paper in radiomics today. It was also published in one of the Nature journals in 2014, and it was from a Belgian group. And it's very well known because it's sort of, first of all, there are lots of data sets. There are 7 data sets, 3 in lung cancer and 2 in head and neck cancer, and so over 1,000 patients. And we will go through their process and see that it's a very strict process to select, that allowed to select a radiomics signature. So this is their workflow. What happened is that first they tested their features, so they extracted over 400 features, and first they tested their features for reproducibility, and both test-retest and multiple observers' reproducibility. So they ranked the parameters according to their robustness, and that was, as I explained earlier, one of the ways to select the most technically robust features. And then they took their first lung population and they did the process that I described. They correlated it to outcome, and they found that a signature with 4 features was very highly correlated to survival. And then they took those 4 features, that's the training set, and they tested if it still worked in the 3 other sets, 1 lung and 2 head and necks, and found that this signature was still significant, still predicted survival in all of these populations. So it was interesting. These are the survival curves. On the left, you have the lung studies survival curves, and on the right, the head and necks. And you see that every single time that radiomics signature separated 2 populations that had different survivals. And it was interesting to notice that these 4 features predicted survival in different localizations. And these patients had different therapies. Some of them had chemo, some of them had radiation therapy, and that this gave an independent information of TNM, of volume of tumor. And I'm not going to show it to you, but it also correlated to gene expression. Now, the only thing is, if you look at the features which are at the top of the graph, you will see that they have funny names that we don't understand. Statistics energy, shape compactness, so we sort of guess what that might mean. Gray level non-uniformity, etc. So that's one of the difficulties in radiomics, is that then you get parameters and you don't really know what they mean. So that will make me move on to the challenges. And the sort of subtitle to this is why, I mean, if radiomics is so great, why hasn't something useful come out yet? Well, because of course it's not quite that simple. First challenge, and I was referring to this, is understanding the results. So for those of you, you probably don't know the man on the left. He's René Descartes, who's actually the symbol of my university. And René Descartes is the one who said, I think, therefore I am, for those of you who are philosophers. It was just to illustrate that we are used to logical concepts. And that's why we like hypothesis-driven research, because once again, we make a hypothesis and we test it. And that's a very usual way to do research. And then we get faced to this data-driven research, and so that's on the right, of course, and I sort of call it the dark side, because things are happening and we don't always understand. First of all, there are some, I mean, we don't know how to do this all by ourselves, all these heat maps and all that. And on top of that, we get features that we don't even know, what their meaning is. So it's a bit, for a lot of us, we sort of feel like we're compromising ourselves. And one of the parameters that we found was significant is the one that I put at the bottom right, which is called GLRLM-RLN. And of course, who knows what that means. Another challenge is to wonder what credit can we give results. So there are several problems about how confident you can be in the results of this data-driven research in radiomics specifically. One of the problems is that there's no standardization. And so people publish different studies, and I'm one of them, but you end up not being able to either compare or reproduce results because everyone is doing things differently. And so there is an initiative which is called the Image Biomarker Standardization Initiative that you can find the whole text at the URL that I put at the bottom right of this slide. These are several people that got together to agree on how to describe features, how to report results, and how to validate the radiomics process. So things will get better, but right now it's still not very standardized. The other problem, and the one that people usually very often talk about, is, am I quantifying a physiological or biological difference, or am I just quantifying differences in images, technical differences in images? And on the right, you're going to see an MRI, a liver MRI, in the same patients, done a few minutes apart, but with different parameters. And just visually, you can see that, you can imagine that the texture that you're going to extract from this will be different when it's exactly the same liver, right? So that, of course, is always a problem. One of the ways that I think we can avoid this is to make sure that our populations are represent a wide variety, both of devices, of acquisitions, of protocols, of institutions, because that way, if a parameter emerges as being significant clinically, then probably it doesn't depend so much on the acquisition technique. But you always have to keep that in mind, because you have to really make sure that it makes, as I wrote in that little blue square, that it makes sense medically, that somehow there's something that makes sense in what we're discovering. The third challenge is the fact that when you're doing data-driven research, then what you're going to discover is very dependent on the training population. And so how much can you generalize whatever you found? And because you might all be getting a bit tired, just for a bit of fun, an illustration, but it does have meaning. Imagine you take an American population and you say, I want to predict what are the 2 biggest factors for celebrity in the United States. And you apply it to the Google images, then you might find that the name Donald and having crazy hair are the 2 factors that will best predict celebrity. But this will be true in an American population of Google images, but of course it might not be true in your own country, and if you're doing this on another series of images, of course. So apart from the fact that I just wanted to add a little humor to this very serious presentation, you just have to keep in fact that the population that you choose will have to be very carefully chosen. And I've also already said this, but I just wanted to remind you that once you've discovered a biomarker using radiomics, you have not finished your work. You still have to validate it clinically. And usually you're going to validate it in the very traditional way. That means that you will have to go through multi-centric trials to prove that when you're measuring this biomarker, it does relate to the clinical outcome. In that sense, you have to decide when you suggest a new test, you have to decide, there's a little schematic here that says you have to decide where your test is going to be. Are you giving this biomarker to replace another measurement, another image test, or another biological sample? Or are you going to do this to triage and then send the people to another imaging technique? Or are you going to first separate the patients and then use this, et cetera, et cetera? And why is this important? This is important in all imaging research. It's important because that's how you're going to choose the population that you're working on. You have to know which population you want to discover something, in which population you want to discover something, so that you choose the right population, and then you start looking for radiomics biomarkers. And so, of course, I'm sorry about the title. I forgot to translate that. You always have to keep in mind the medical meaning. Where does the parameter I'm discovering fit in the patient management? And you have to think about its actionability. So is it actionable? What does that mean? That means, okay, I discovered it, but will it mean something for me to use it in the patient management? For example, if you find a new biomarker to predict a response to a therapy for which there is no alternative, then there's really no purpose to discover it, right? So you'll have a nice biomarker, but your oncologist will keep on giving the same therapy because they have nothing else to give. Or you will, if you predict an outcome that has, of course, survival is usually pretty interesting, but there might be other outcomes, or predicting a gene expression that that doesn't alter therapy, or things like that. I just wanted to remind on this slide that we have to be humble, but we shouldn't be, afraid or feel that our discoveries are not always actionable, because in genomics, for example, there are less than 1% of the discoveries that are actionable. That means that lead to a change in patient management. So I think we have to realize that probably in radiomics it will be similar, and we will make a lot of discoveries, but not that many will lead to changes in our clinical routine. The last thing I wanted to underline was the phenomena that goes with data-driven research, which is what I call the data rush. There was a fight for fire, then there was the gold rush, and then there was the black gold rush, the oil rush, and now there's a data rush. Why? Because everything centers on having the data. And that is a very big challenge, because we all have packs and we all have a lot of patients with a lot of images, but it's just not the same thing having the images and being able to use them. Because what we need are clean and annotated cohorts of patients. And it's kind of frustrating, because when we read our radiological images, we are annotating all the time. We are measuring things. We are sometimes, I just drew a perfusion curve that I measure on an ovarian cancer, for example. And yet most of these annotations are lost because we cannot export them from our packs. So that is really, for me, a very important part of what we need to be working on to make our annotations effective, to actually put together cohorts. Because when you have to go back and annotate all the patients yourself, well, then you don't have enough energy to do them all, and you end up with several tens of patients instead of having hundreds of them. The other source of information are our reports, because we do radiological reports all the time. And in our reports, there's lots of information. Right here, I guess it doesn't really go with the images I put underneath, but I put a report which is in French, of course, because it's one of mine, but about a fibroid. And when I reread my report, I realized there was so much information about the size, where it was, all the signal on the different sequences. There was even a quantitative ADC value, and there was a diagnosis. And these reports are unfortunately free text. So there's a lot of work going on, but we also have to work on the extraction of all this information for our reports. So basically what I'm saying is that the data is there, but right now we don't have the tools to extract all this annotation and all this information in a very effective way. So just to start concluding, why should I do radiomics? I do radiomics so that I can test simultaneously a large number of new parameters extracted from my images. And so I will hopefully discover new potential biomarkers that will then need to be understood and tested for validation. It may also help me generate new hypotheses because I might do a heat map, discover new phenotypes, which will allow me to go back on the patients and maybe discover new molecular profiles. But for that, remember, we need data. So we really have to work on having cohorts. So how do you start if you want to do radiomics? Well, there are lots of open source software that you can download. It's a bit, of course, you have to sort of work on it because there are quite a few things to understand about how these textures work and eventually pre-processing that are applied, because otherwise you're going to start extracting things that don't really have any meaning. And then the last message is that if you're a radiologist all alone, then you might feel like Charlie Chaplin looking for gold all by himself in his street clothes. And you can't really do radiomics all alone. You need a whole team around you. And that's just a nice occasion to make new friends, data scientists, medical informatics people, statisticians. So I find that it is very enjoyable, but it's really a group collaboration. So thank you very much for listening. I'm now finished my presentation and it is open for questions. So please put your questions in the Q&A box and I will then do my best to answer them. Thank you very much.
Wow, that was amazing. Really very impressive presentation. I probably, until the viewers have posed some questions, I can ask some questions myself. So I was wondering if you went a bit through the pre-processing step. Could you expand a bit on that as well?
Yes, I didn't talk about that at all. Actually, you usually to extract features, you can't just do it on the data directly. It actually depends sort of on the type of features, but for example, shape features, you can do it directly on the data. But when you're doing texture features, for example, you actually have to aggregate your pixels and aggregate your gray levels because otherwise, so I was saying you want to see if a pixel is surrounded by pixels that are similar. And so you have to define what similar is. And if you keep your original data, for example, in CT, well, you can have 2,000 different levels of gray levels. Right, and so there will practically never be two pixels that have the same value. So you're going to say, I want to aggregate them by 10 or 20 Hounsfield units.
Yeah, right.
And those are pixels that are similar.
Okay.
Yeah, that's the gray level aggregation. And then you'll aggregate the pixels so that you don't have tens of thousands of pixels because that would take longer to compute, for example.
All right. And so you work basically mainly in CT. Did I understand that right?
Well, what I presented is on CT, but we're also working on MRI. And actually, it is quite different. The pre-processing that you're going to do is different as well. I don't know if you want me to expand on that.
Yeah, if you don't mind, because until now there's no answer, no questions from the audience.
Sure. So CT is a bit more straightforward, particularly regarding the gray levels, because as we know, gray levels are absolute levels of linked to the attenuation of tissues.
Right.
Whereas in MRI, of course, they're relative signal intensity. So if you aggregate them, first of all, if you aggregate them, you might end up with differences simply because the range of signal intensities that you have in your image are too different. And so you might conclude that the two images are very different, whereas they aren't really, or vice versa, of course. So actually, what I work on a lot with my students is not so much the clinical relevance right now, because we sort of noticed that there were technical issues on how you calculate these features. So we're working a lot about that, and one of my master's students is testing all the different ways to aggregate on MRI and how it impacts the calculation of the features. But it's definitely a big issue. They talk about it in that initiative that I showed you, the IPSI initiative.
Yeah, right.
but actually they stay kind of a bit conservative. Well, they don't really take a stand on MRI. So, yeah.
Yeah, that was actually what we noticed here as well, because there seems to be no consensus on how to handle MRI data so far.
Yeah.
Actually, for MRI, also, do you usually use just one sequence like 3DT1, or you can use several sequences and so increase the number of features that you can extract?
Okay, thanks a lot for that question, Nadia, because we're actually submitting a paper about that. It should actually, I hope it's submitted. Yeah, we worked on whether adding several sequences added extra information, or if you just sort of repeated the same information. And interestingly, the different sequences give different information. Some of the information is in common, of course, but a lot of it is different. And even what I found very interesting is even different sequences that come from the same Dixon sequence. So for example, in phase and water, even though they come from the exact same images and there's just a mathematical calculation, right, that generates one of the sequences and the other, they actually do give different information as well. So I think it's what we consider is that in MRI, you ideally, it enriches the information to add several sequences.
Thank you. And also I have a question about the training population. So how big should be the training population? Is there a minimal size of training population below which the results are not robust enough, or it will depend on the features that you want to get?
Actually, once if you go through feature reduction, which is what we did, then the usual laws of statistics applies, right? So it's just like any other study. You know that if you're under 30, then probably it's a bit difficult to make definite conclusions. And also theoretically, you're supposed to have 10 times more samples than features, right? That's the theory. In reality, it's practically never the case. And if we refer to genomics, which is what we refer to when we do radiomics, because everything that I showed is inspired from genomics, then they are never in that situation because they have like tens of thousands of genes. So I think what you have to know is that there will be a high level of false discovery rate. That's just something you have to accept. And that's the point. Of course, it would be better if we had giant populations, but most of the time we don't. So we just, that's why you have to understand that radiomics is a discovery process. You discover, but you don't validate. But the idea is that instead of doing lots of studies and testing biomarkers and none of them work, and doing very time-consuming and money-consuming studies, well, you're going to select them and only put your effort in those selected biomarkers, and those you're going to invest a lot in it to make a big population, to do a multi-center trial, etc. Was that okay, the answer?
Oh, yes, thank you. So everybody can try to do it, even if he doesn't have these huge populations finally, but then we have to be careful about our results, basically.
Exactly, basically. And I don't know if I showed two survival curves. Actually, I went very fast on that because you also see the error curves, right? You see them on both sides, and they're actually very large because our population was not, we have two populations and they're under 100. So the error is large. We have something very significant, but that means that we know that we can invest time to work on those parameters.
Yeah, thank you. And another question I have, so you talked about all these softwares that are available that we can try to use. Do you really need data scientists to help you using the softwares, or radiologists can use them if they're careful about what they're doing with?
I think a radiologist who's done image processing a little will be able to understand them. The only issue is that I can change the slide, I guess, if you want to see which ones. I mean, these are just 3 examples. I'm just showing them because they're open source. And there's a French one, so we're proud. But the issue is that you might end up with absurd results if you don't really understand what's going on. And just to give the example, that's what. So we worked a lot on CT. We have our own software because these did not exist when we started. And then we started trying these out as well. And for CT, it worked pretty well. But then when we went to MRI, we got really weird results. And that's why we went into understanding about pixel aggregation, gray level aggregation, etc. And so if you can't have that critical view of these software, then you might end up just having absurd results. So I think you have to have at least an experience in image processing.
Okay, and the best thing, as you told, is to work together with the engineers in image processing.
Exactly.
Yeah, very interesting. So just probably one final question that I would like to know your views on is, so we heard there's going to be lots of false findings maybe. Is there any like roadmap you would envision for the next steps to bring radiomics to really have an impact in clinical routine?
A part of this is covered by the IPSI initiative. So because they are really working on standardization, there's also a radiomics quality score that was published, which sort of is supposed to help you score the quality of the study. Mm-hmm. So as things will go, first of all, I think that it will get little by little harder to publish small radiomics studies, obviously, and low quality radiomics studies. So we'll have to increase the quality. We definitely need both training and validation data sets. I don't think a single paper will be accepted if you don't have at least a validation data set. But in the roadmap, I think one of the important things, if we want it to be clinically relevant, are some of the issues that I showed, and particularly asking the right question and choosing the right population. Because I think that sometimes we miss that, and I'm including myself, and this is not specific to radiomics. And I like to give the example of studies that I've done so that I can embarrass myself. For example, on large rectal cancers, and we work on, or large cervical cancers, and we work on response to chemoradiation therapy. And we showed that with perfusion you could predict. And then when we told our oncologists and the tumor board, they said, okay, but if you tell me that he's not going to respond, what else can I do? I can't operate him, he can't have surgery. So what are you telling me, not to treat him? And so suddenly I realized that I had a really interesting biomarker, but, there's no clinical point because we have to treat the patients with this, and we have to give them the best chance to use this therapy. So I think that we have to understand that, and we have to understand what the clinical question is and using it on the right population.
Right. Yeah, very, very important. Very nice overview. Actually, it was a very nice presentation. I liked it very much.
Thank you.
Thank you very much for the presentation.
So far, it seems there is no questions from the audience. So I don't know. Is there any?
I don't see any. No.
Okay.
Okay, so everything was very clear for the audience.
That's good. Yeah, that's good.
Yeah.
Okay.
Then
I guess we'll see if we close the webinar or we wait another couple of minutes.
Well, I think if there are no more questions, we can close the webinar. So thank you everybody, for all the audience, for assisting in the webinar, and thank you very much, Laure, for this interesting and complete presentation.
Thank you very much, and have a nice evening.
Thank you. You too. Have a nice evening. Bye.